Boa tarde.
Good afternoon, everyone.
So we'll start now the third panel of the 6th International Symposium of Malologicals. I have the pleasure to receive here Dr. Eduardo Estrada and Dr. Andreas Castan. Just to give you an overview, some recommendations regarding the introduction of some questions. We ask the online participants to bring the questions by the chat in the application, the IPP. And here, the people who are here, they can bring written questions for the ladies that are here, so we can bring for the presenters, okay? So first of all, I would like to present Mr. Eduardo Estrada. He is chemical bacteriologist and parasitologist, graduated in Mexico on the National School of Biological Sciences, has more than 25 years working in the pharmaceutical industry, rich experience in Pfizer and Sanofi, especially in the last 15 years. So, Mr. Eduardo Estrada, first of all, thank you for being here, collaborating for this symposium, and please feel free to share your knowledge or experience on the topic. Thank you so much.
Good evening. Hello, everyone. Before diving in my presentation, I would like to thank the organizers for inviting me to the symposium. I'm glad to be here sharing my experience in the pharmaceutical industry and in the biologics production. I divided my presentation in different sections, and the first part is to talk about the vaccine specificities. The second one is to talk about the current production systems or models for vaccines. And the third topic to cover in the session today is about to talk about some items, how the vaccines manufacturing sites will be in the future. And last, not least, is to discuss about some items that we consider that should be taken into account for the future starting operations in a new site. So, starting with the specificities of vaccines, we know that vaccines are sophisticated products, difficult sometimes for characterization, because as we know, they are produced, the vaccines, from live microorganisms. Their production is a long and complex process because using live microorganisms, we need, or the facilities, in the design of facilities, we need to consider special buildings for containment. Additionally, we know that the production vaccines are under regulated organizations have in place different specifications, making difficult the process for registration, for changes post-approval, and the release. So the next point, so how the vaccines have been produced since the past, since several years ago up to now. The classical model that we have been followed is to have facilities tailored for specific products, but not adapted to face the challenge that we will have tomorrow. Sometimes the management of the resources are quite difficult in terms of hand labor or qualified people and so on. But for that, there are some observed challenges that the companies face day to day. For example, we have long and lasting costly in terms of construction new facilities or expanding existing one. And the other point is, as I mentioned, the long cycle management of the vaccines. Because as we know, the vaccines production, the batches sometimes are small, the viability of in the production is high, difficulting the release in many cases. Another point is that it's quite difficult to reconvert or to expand existing facilities because there is not enough space. The cost for that is high, and all of that is a complex process currently in the manufacturing process of vaccines. But for that, we have different alternatives. One alternative is to expand, of course, the existing facilities. Another one is to work additional capacity with external companies, working with contractors or with partners. Another point is, could be the best solution, but not always is possible, is the construction of new buildings, integrating innovative technologies in a single facility. But when we have unsolved challenge, of course, the consequence is to miss supply opportunities and increasing the cost of production in many cases. Of course, the current approach is less flexible for vaccine production. In the next slide, I would like to say that the vaccine manufacturers have been warning, looking for vaccines development with new technologies to meet the demand. The last 2 years learned that the companies, most of the companies, were not prepared to meet the demand for new vaccines, new capacity, and the tech transfer between companies increased a lot during the last 2 years. So the question is, how to meet the demand of the vaccine, being flexible, being agile, in order to meet the customer requirements. For that, there are some items that we have considered. The first one is to have a flexibility and modularity in the same site, where the manufacturers can produce at the same time or simultaneously different products and with different characteristics, being flexible in the changes of the products. Additional item is considering modular docking stations, single-use technology, and the use of the space, enough space in order to have more flexibility in the operations, avoiding cross-contamination. The other point is the use of the autonomous or the robots or cobots. This is not, sometimes this sounds like it's not, it will be in the future, in the long term, but it's not the case nowadays in the pharma industry. For example, the use of robots or cobots can reduce the cross-contamination, can reduce the hand labor, avoiding connection, error, compliance, and so on. This is helpful when we have complex process, and in order to simplify the cycle time or the change management in the products, can be used cobots and robots. Another important point is that how to monitoring on real time the process. We can monitoring on real time having KPI in performance indicators in terms of quality, compliance, manufacturing. If we use digital tools, for example, connected mobile devices in the phones, you can monitoring on real time the process, the KPIs, the complaints, the change control, everything in the company. Additionally, digital schedules, data analytics, you can monitoring on real time how the analytical is being, how the results are on the products. Another one is about, as I mentioned, digital, to simulate in anticipating any risk or anticipating how to make the best decision in order to avoid any failure. The other one, in order to be aligned with environment, is to reduce the energy consumption using energy recovery panels. This is very important because we should consider all different times in order to be aligned with regulations. And another point is virtual reality simultaneous training. For the operators, it's very important because they want to simulate and receive real training using virtual reality for a specific training, for working in aseptic operations, for example, or working in a complex steps of the manufacturing production. It's very useful to have virtual reality for training, specifically for when for newcomers in the companies. So we can say that new manufacturing asset should be sustainable, agile, and flexible, and the vaccine business requires continuous massive investment. This is critical because, of course, moving from the traditional or native systems for manufacturing vaccine, manufacturing, moving to the future for new facilities, state of the art, we need to invest. We need to invest in new technologies, in new platforms, in order to reduce cost of products, in order to be agile to meet the demand of the vaccines, and of course, being in compliance with regulators and with internal procedures. And the next one, and the last point of my presentation, is some technical and soft skills that we should consider operating new facility. Of course, this message can be adapted depending on local or global context. The first one is to consider that we should have technical knowledge and industry experience. This because, as we know, following with the next item, we have to act for change because last 2 years learned a lot that we were not prepared to face the pandemic. At the beginning, of course, was a tremendous worry about the situation. So we should be prepared to act for change, for new technology, for new facilities, for new regulations, for any requirements that the environment demand. The next one is strive for results. What results we need to focus our operations in a new facility, in a new process, in a new product. The next one is to cooperate transversally between companies. As I mentioned, during last 2 years, the tech transfer exercise increased substantially, and the cooperation has been essential in order to be ready with the products or to be ready on time with the new vaccines. So the cooperation, the communication between intercompany or between companies with contractors, with partners, or with regulators in a transversally cooperation. The other one is commit to customer. At the end, the benefits of the new facilities, the new products, the new platforms will be the customers. The customers are our patients, our regulators that we need to be in compliance and to satisfy the demand of their requirements. The next one is to think in the future, having a strategy to face the challenge to face tomorrow or in the future with new technologies and new platforms. The next one is how to lead teams. Lead teams, I mean, internally or externally with regulators, with partners, with contractors, that we need lead teams, of course, to satisfy the demand. And the last one is, and another one, sorry, is develop people. Develop people in the symposium had a lot of presentation talking about new technologies and new platforms, new vaccines. And develop people is essential, is crucial to be updated. This symposium is an important forum to receive or to exchange innovation and information about people, about new technologies, and of course, increasing the people development. And the last one is a make decision. The decisions, if we define to go with a partner to build a new facility, but the new facility to make the decision to invest in a new building, of course, requires a massive investment that at the end, with the state-of-the-art, with new technologies, the product will be produced in an agile way. With agility, the site will be flexible to produce different products simultaneously to satisfy the demand. Okay. Thank you.
Thank you, Dr. Estrada. Just to give some more information regarding the questions, the online participants should bring the questions by the chat on the application, and the in-person participants can also use the chat, or you can raise the hands so the ladies here can support you to bring the written questions. Okay, so I'll pass the opportunity to Mr. Andreas Kastan, Mr. Andreas Kasten, who is online. He has been working on the biotech industry for more than 20 years. Right now, he's working, providing leadership and support to Citiva Biopress business. So I invite Mr. Andreas Kasten to share his experience in new technologies and strategies in biopharmaceutical manufacturers. So welcome, Mr. Kasten. Thank you so much.
Yes, thank you very much for having me. Good afternoon, everybody. And thank you very much for the nice introduction. I would like to speak about new technologies in the biopharma industry, and I hope in my talk I can convince you about that the future of process development is in silico. But before I start, I want to give you an overview of my view of the industry, which is very well aligned with the BPOG roadmap. And this is my flavor of that roadmap, where the market is seeing an immense growth. We are seeing new modalities coming up. We are seeing an uncertainty in clinical outcomes and demand projections, but we are also seeing an immense cost pressure on the industry, and this drives the business towards building flexible facilities, and we heard that in the previous talk, a drive to better quality or a focus on quality, and a drive for speed to clinic, speed to market, and an immense pressure on cost reduction. So what can we do to increase the speed through the CMC journey? And I would like to start with just drawing the map of what this CMC journey is about. It's about moving a process, a project from early PD to clinical manufacturing, back to late PD, into the MSAT group and to commercial manufacturing, maybe outsourcing it to a CDMO. So this journey is really complex. This this is a quote from a paper in biotechnology bioengineering, a paper authored by David Rausch and many co-authors, and it says that in the future it may be possible to perform most development activities in silico based on data and predictive models. And that is kind of the leading star for my talk and where the industry is moving towards. But I also want to start by giving you the different flavors of modeling approaches, the different modeling paradigms, starting with the first principle models, where you have a huge process understanding and very little need to calibrate the models, the mechanistic models, which still describe kind of your process understanding with a higher need for parameters, moving into the statistical models, where you basically do a statistic fit of your data, requiring more data and less insights. And on the other end of the spectrum is the machine learning and artificial intelligence, which basically requires no insights and a huge amount of data to go into the models. We asked our customers, or did a survey about what are the desired outcomes when running these simulations, and looking at the kind of top 9 responses that we received, these can be condensated into a desire to increase the process robustness, to increase the scientific understanding, and again to accelerate the CMC journey to increase the speed. Speaking about the understanding, I want to highlight how in silico models can increase understanding in this slide, where we have the one factor at a time experimental approach to the left, with very little in-depth process understanding. We have the classical DOE in the middle, which gives more understanding of the process and the process parameters, and this can be further increased by combining it with in silico modeling of the process and gaining even more insights. So where can in silico process development have an impact? And my answer is basically everywhere, starting in early stage in the definition of the process, and I will show a case study on that later on. In the late stage, in the optimization, in the process characterization, in the commercial, in improving the process and managing change, in the process transfer and scale up. And again, here I have a case study. And in the manufacturing stage, in training the operators, and the previous speaker elaborated on this as well as in the commercial setting, in implementing soft sensors, adaptive control, and planning the operations. There's also an expectation from the regulators to implement these kind of virtual simulations to increase process understanding and solve problems at the manufacturing scale, and this is coming from an FDA document. But let's shift gears and look into the case study that I promised. The first one is about bioreactor scaling, how we have built in silico models to facilitate bioreactor scaling. But let me start by speaking a few words about what bioreactor scaling is actually about. It's very much about setting the aeration and agitation settings so that the cellular environment for the cells is kept constant across different bioreactor scales, because the cellular environment allows the cells to grow as expected at the viability, produce the product in the quality that is expected. And here I draw a map of the variables, the parameters defining a process, and there are the ones to the left, which are very easy to scale, or basically are not scaled at all, the temperature and pressure. You can volumetrically scale the nutrient additions. Coming to the right, it's more difficult because these parameters do not scale volumetrically, like the bubble sizes, the size of the cells. There you need to have more advanced tools that help you navigating in the design space of dissolved oxygen control. And I try to depict this or describe this design space multidimensional to the left, where this donut describes the design space and you see the parameter describing it. I try to illustrate this a little better in a projection into the two-dimensional space, where we have the agitation on the y-axis and the aeration to the x-axis, where to the left, lower left, we end in a situation of mass transfer limitations. To the right, with high aerations, we see foaming and bubble damage issues, and to the upper limit, we see hydrodynamic shear limit damage. and in the middle is the safe design space, which we need to describe and also find a design space across different scales. So what are the in silico components going into the bioreactor scaling? It's of course doing the homework of finding the equation that describe the oxygen transfer rate, knowing the parameters that impact the model constant, which in this case is the KLA. You need to set up experiments to build empirical DOE models, or you move into in silico, that is CFD models, to derive these models for the KLA. The next step is then to combine these models for different sizes of bioreactors, microbioreactors, benchtop, as well as production bioreactors, and then apply the models to translate operating parameters from one scale into the other, but also upfront looking at the design space in the different scales overlapping, and by that means ensuring a smooth transfer across scales. On the next slide, I have some customer data from UMass transferring a AAV production process in HEC-293 cells from a 10 liter to a 200 liter bioreactor. And to the left is a snapshot of the outputs of the scaling tool. And you see the parameters that were kept constant in the scaling, which is the power density, the volumetric gas flow, and the oxygen transfer. But in the right panel, you see the readout from the two processes, in green the 10 liter and in blue the 200 liter, where you see pretty comparable cell densities and viabilities. I would even say improved performance on the 200 liter scale. And the customer reported back that using the tool removed the guesswork and achieved a consistent run at the first try, producing this very precious material. Now I want to shift gears and move into the next case study, which is in building an in silico model for chromatography step development. And again, the first step is really understanding what are the kind of components in this approach. And it starts again with the understanding the process, the convection, the dispersion, the diffusion, the mass transfer inside the bead, the interaction between the different ligands and the target protein, translating that into differential equations, first order or mechanistic equations that describe these processes, and then feeding or fitting the equations with column data, system data, and a limited amount of chromatography data, and by that enabling the simulations of thousands of process conditions within a few hours, allowing each simulation basically taking between 5 and 10 seconds, and allowing a generation of a large amount of data. And in the next slide is a comparison between a traditional DOE approach, seeing the two parameters, ionic strength and load density, a classical DOE with 9 experiments, each experiment taking about 4 to 8 hours, translating in a runtime of 1 to 2 weeks. Comparing this with the model-based design space, running 1,000 to 10,000 in silico experiments overnight, and describing the full design space here, even allowing an extrapolation outside this calibrated space. And in a recent publication from Boehringer Ingelheim, it is claimed that the in silico approach allowed a reduction of the lab experiments by 75% in a process characterization study, which is of course significant. But let's look at chromatography study comparing a DOE optimized process with an in silico optimized process, where the objective was to separate the target protein from a low molecular species, giving a high purity target. However, the DOE optimized process resulted in a comparably low yield. So a lot of material needed to be eluted together with the low molecular species, whereas the in silico optimized process achieved the same purity, but a significantly improved yield in this step by adjusting the gradient, adjusting the low density and the column length in this run. So what can you do with these models? You can optimize your process, and I think I showed you that on the previous slide. You can use the model for tech transfer to predict the conditions when moving into different facilities, which maybe doesn't have the ideal column hardware available, facilitate scale up, but also different load conditions. It can help you in the risk management, that is in identifying the critical region in your design space where you then could put your experimental efforts and reduce that significantly. And you can use it in deviation management, in looking into troubleshooting manufacturing, identifying root cause, and in summary, mechanistic modeling really helps you to digitalize the process development. Again, a quote from Boehringer Ingelheim, which says that the move into in silico modeling is the largest game changer in over 20 years, increases understanding and takes down the effort and cost in PD. So what would a journey look like for a pharmaceutical company? The first step obviously is to identify the use case. What are the low-hanging fruit? What would be the first use case? Start using the tools that, for example, the ones I described today, and then implement these to start with in the PD to optimize your process, and then in the next step in the tech transfer into manufacturing. It's a different way of working, and we as Cytiva are definitely here to help our customers in this journey. So I hope I have been able to convince you that process development is going in silico. Modeling will be used to accelerate the CMC journey. New in silico platforms will emerge. I showed you two examples. We have more in our pipelines, and the gains are very well aligned with the tech roadmap I showed you in the beginning, as well as the expectations from the customer survey. That is increased robustness, increase process understanding, increase speed, increase the performance of the process, and take down the cost. And with that, I want to thank you for your attention, and I'm very happy and looking forward to the discussion.
Thank you. Mr. Castan, thank you so much for sharing your experience with us. It's a pleasure to receive you here. Up to now, we don't have any question. I don't know if the in-person audience has any question. I have one, Mr. Estrada. Once we are talking about agility in complex scenarios, that's what we are facing for the last 2 years. What you consider, according to experience, the most important legacy that this pandemic left to the production network we have right now?
As I mentioned during the presentation, different items should be considered, but to me, or in my experience from last 2 years, is to be prepared for the changes, because the change in the manufacturing, in the environment, nowadays, the way we work is different. The companies adapt their process, their systems to continuous operating within a confused environment, uncertainty environment. So to me, anticipate the changes, having or improving the knowledge of the environment can help to anticipate risk or to anticipate failures in the manufacturing vaccines. Yeah. To me, is act for change, to anticipate the change. And the other point is the communication or to identify additional capacities or rethinking in the current capacity, or to be prepared to expand capacity with contractors, with new facilities, with new technologies and new platforms. This is key in order to be prepared and to anticipate any potential adverse event.
Thank you. We have a question here from the application from Sebastian Machado Weber. The question is it planned from Sanofi to have manufacturing sites in Latin America to reduce costs?
In Latin America, currently Sanofi has 2 facilities, one in Mexico, operating one in Mexico, another one in Argentina. In Argentina, producing antigen for hepatitis B, and in Mexico, antigens for flu. And in expansion, yes, in Latin America, in Mexico, is under construction and qualification a new facility for formulation, fill and pack flu vaccines. This facility, the last one that is under construction and qualification, have a new technology on their implementation process for virtual training, as I mentioned in my presentation, useful for newcomers and for training of the manufacturing operators, but not additional, from my knowledge, no additional facilities in Latin America.
Thank you. Thank you so much. Okay, we have one more question.
Thank you both for the very exciting presentations. We are entering in a new world. And my question for Dr. Andres, and also Dr. Estrada, please feel free if you want to add something. But my question is, do you think that the national regulatory agencies are prepared to receive in silico validation results or in silico results for change control, for instance? Do you think that we are prepared to this, to enter in this new world with the national regulatory agencies are able to receive this kind of documentation?
I didn't catch the last part of your question. Could you please repeat it?
The last part is if you, both of you think that the national regulatory agencies are able to receive this kind of new world documentation. What I mean, in silico simulations of validations or post-change, post-approval changes, so simulated in silico. That's my point.
Yeah. I can start answering, and my perspective is that I mean, I showed you the quote from the FDA, so there definitely is a willingness and almost an expectation to facilitate this type of filing, and what I have seen from my own experience is that very simple chromatography steps like ion exchange step, for example, that has been backed up by solid mechanistic models and have been as a kind of test case filed solely by based on the simulations rather than making the process characterization studies. I think when these simple steps are established, there's still a long way to go for more complex unit operations. Just thinking of a mixed mode chromatography, I think is still a stretch goal, but I think the industry is moving towards these steps, and like I just described, also use the models to guide the experimental effort into a region where criticality is expected, and rely on the in silico data in the rest of the science base.
Dr. Estrada, would you like to add some comments?
Yeah. My point of view for your question is that we need to increase the communication with regulators in order to cooperate, the transversal cooperation that I mentioned, because the new ways of working, the new system, electronic system, like electronic batch, for example, the new digital tools with the information provided, how to simplify the release process, all of these new initiatives should be shared with the health authorities, with the regulators, in order to be in compliance, but simplifying. We learned that of the last 2 years, the approval of the vaccines in record time. So we should increase the communication with the regulator, showing the benefits of the digital tools, or the new systems, the new technology, the virtual information that we can provide in order to grow together in benefits of the patients. But of course, being in compliance with the regulatory requirements. But to me, the important point is to improve the communication with the regulators, sharing this kind of new ways of working.
Thank you so much. The last question from Aline Oliveira. First talk, Dr. Estrada. The question is, to make process development in silica of biological products, how do you simulate the cell metabolism and yield?
The same metabolism. Yes, of course, talking about the simulation, with the new tools that, as I explained, can help to simulate with using virtual tools, simulating situations for making decisions in different scenarios. And of course, with this, we can reduce the human error, we can reduce the number of deviations or changes or failures or mitigating or the risk mitigation. This can help to for all of that. And of course, this is real in some companies that are starting using this kind of tools. But of course, can be do with this kind of virtual tools or digital systems.
Thank you so much. One more time, I'd like to thank you, Mr. Estrada. Thank you, Mr. Castan, for sharing your experience with all the audience here. Thank you so much. Thank you.
My pleasure. Thank you for having me.